Okay, thank you. Now I think everyone has joined the lesson,
Yeah,
the
yeah.
the platform. Okay. So today I will talk about the genetic diversity in pre-crops in terms of assessment, exploitation, and conservation. Just an overview of these three aspects. So the content will be how we can assess genetic diversity through molecular markers and their use, exploitation of genetic diversity, and so we will see some principle for genome-wide association analysis, and also what has been done in pitch for in a large GenPlus collection in pitch for diversity assessment and GWAS analysis. And we will end up with some aspects of the conservation of the genetic variability. So, what is genetic diversity? We already know genetic diversity is at the very basis of the three level of organization of the whole concept of biodiversity. And it refers to the diversity within different genotypes of the same species. In crops, genetic diversity refers to the variation of inheritable genetic traits within and between population of a plant species, and it encompasses the presence of different alleles at specific loci. Genetic diversity assessment is done through the use of molecular markers, mainly microsatellite markers or simple sequence repeats, SSRs, and SNPs. Microsatellite markers are short tandem repeats, stretches of tandem repetition of core motif at one locus. They are codominant, mostly single locus, are highly variable, so we can find many alleles per locus, in which we can have up to 18 alleles. Of course, in one diploid individuals, only 2 of them are found. Then we have they are transportable across related species. And they have a low to medium throughput. In fact, we can multiplex them with appropriate design of combination of markers. We can combine up to 10 microsatellites. There are a lot of resources of markers, microsatellites developed in all the main crop species. So I didn't report here a comprehensive list for fruit species, but just I report here the last set that was identified in after the genome sequencing of the pitch genome that are long core markers that were developed in pitch, but are also useful for analysis on the other 4 diploid prune species: almond, apricot, Japanese plum, and sweet cherry. They prove to be effective for individual characterization, parentage, and population structure analysis. Okay, then we have single nucleotide polymorphisms, also known as SNPs. These are variations at the level of a single DNA base. So they are biallelic and codominant, so less polymorphic informative than microsatellites, but are the most abundant in the genomes. So it's possible to perform high throughput analysis of thousands of markers per sample in one reaction. Also for SNPs, there are different, many platforms have been developed, many resources have been developed, either as arrays of SNPs that go with hybridization, that were developed in apple, peach, strawberry, cherry, grape, and almond. But we can also directly detect single nucleotide variations through techniques based on next generation sequencing. One of these is genotyping by sequencing, which is based on the sequencing of restriction digested DNA fragments. And also, more recently, there are also targeted genotyping panels which work similarly to GBS, but on targeted specific loci. And these are available for grape, peach, apricot, and lemon. Yes, so with molecular data, we are able to measure genetic diversity. Now, I won't go through all this list, which is a list of some of the indices and statistics that are able, that are with which we can analyze our material through molecular data. But just to say that, or by row counting of the alleles, or through the analysis of allelic frequency, we are able to analyze and compare samples within a populations, or the variability present between populations. Another way that we can use and visualize our market data, molecular data, is through the cluster analysis, which is a multivariate statistical technique that groups accessions into cluster based on a similarity matrix, which is calculated from pairwise similarities between all individuals. This gives a visual representation of the genetic diversity present in the germplasm collection, and identifies relatedness among accessions. Here you see a dendrogram, which is a way that we visualize cluster analysis results, where samples are plotted in the dendrogram. Similarities are plotted from the left side to the right side of the dendrogram, with increasing values, so that at the end samples are either separated, or those that are identical are still linked by these vertical bars. So we can analyze our collection with cluster analysis to verify true to type accessions, like you see in this case, where this is a detail of a much larger dendrogram. You find these two genotypes that were clusterized with the cultivar reference with the same name, so they are true to type individuals. Or we can also find some errors in the collections like synonyms that are when accessions with different names have the same genetic profile, like in this cluster where the cultivar Capo d'Asino is the cluster of this cultivar, and we find a fragola, which is obviously a synonym. On the other way around, we can also detect varieties which with the same name that goes in different clusters, so so-called homonyms. Here are some software resources. There are many resources to do this kind of analysis. Here are just a few of them. If anyone wants to try to, some of them are free, can be downloaded from the internet, and some other are like Genome Studio and CLC Genomic Workbench are need a license. But there are many other resources so to make this kind of analysis. Okay, so just a brief overview of what we can do with molecular markers, and we can assess the genetic diversity. And now we go through the exploitation of genetic diversity through genetic approaches. In this case, I will give you some principles of the genome-wide association analysis approach. GWAS is a genetic approach that can identify association between genetic variations, so polymorphism, across the entire genome and the traits of interest in natural populations. It requires high-throughput genotyping, so genotyping through SNPs, and also phenotyping of all the individuals of the population. And it is based on association mapping. Association mapping takes advantage of the recombination events during the evolutionary history of a species, so that you see that after many generations, many cycles of meiosis, initial genomes are shuffled and divided into blocks, so that a gene of interest is co-segregates with the block of the starting genome. What is linkage? This is based on linkage. Association mapping is based on linkage disequilibrium. What is linkage disequilibrium is the non-random association of alleles at different loci on a chromosome. So if we have two genes with two alleles, and there is complete equilibrium, so no association, the four alleles are segregated randomly and we independently, and we can find all the classes of genotypes. While when there is association, in this case complete association, only combination, particular combination of the alleles are inherited, tend to be inherited together. So we find different in the allelic frequency. In this case, we don't observe, we cannot observe this combination, or the combination big A little b. So how LD association mapping leverages LD for association analysis. In a GWAS experiment, we don't genotype all the SNPs. Of course, it would be unfeasible. So in this case, a proportion of the genetic variants is not genotyped. But in this situation, we can have two different scenarios. We have the genotype, the variant that is linked to the trait, that is causes the trait, that is involved in the trait, which is also that has been genotyped. In this case, we have a direct association between the genotype and the phenotype, and we associate the causal variant. While when we, in case of when we genotype, the causal variant is not genotyped, we can make the association of a phenotype with the region of high linkage disequilibrium through the genotype that lies in the linkage disequilibrium region. The level of LD is essential for designing an association mapping experiment. In fact, you see here that if LD is high, blocks of linkage disequilibrium are larger, so we need less marker to characterize and to target each block and to target the gene of interest. In this case, we should have more meiotic events, so more individuals. While in the case of low LD, we should need more markers to target each block, LD block, and to so target our gene of interest. In this case, so we need less number of individuals, but we need a higher number of markers. Sometimes this situation is too the statistical power and the statistical calculation power is not enough to do a genome-wide association analysis, and only a candidate gene analysis can be performed. So in the very essential step, GWAS analysis workflow is illustrated in this slide. So we have our germplasm starting panel of collection. We genotype them with SNP markers. So we do a high throughput genotyping of the material. Through the genotyping, we can analyze our material to check for population structure and for evaluating linkage disequilibrium. In the meanwhile, we must phenotype all the accessions of the starting panel. Then we put together the two type of information to have likely at the end the identification of a market trait association. Now I will. I want to illustrate what was done. This concept were applied in a germplasm collection of peach, in a large germplasm collection of peach. This was the work that was done in a previous European project from the past Horizon 2020 framework that was called Fruit Breedomics. in this project, we had 6 germplasm collections. So we added up the materials from 6 germplasm collections, 2 from Italy, 1 from CREA, 1 from Spain, 2 from France, and 1 from China, to sum up 1,580 prunus accessions. They were mainly entirely, almost totally prunus persica accessions. And we genotyped all of them with pitch SNP pitch array of nominal 9,000 SNPs, 9,000 SNPs. Okay, the first step in when you do an hybridization with the molecular SNP array, you have to filter the panel of the starting panel of markers. So we filtered out those markers giving ambiguous signals for the presence of null allele or preferential annealing, those with duplicated sequences and genes, those which were not were in fact markers because were monomorphic, didn't detect any variant, and those that failed to give any signal. So we ended up from the starting panel with 5,330 markers. These markers were used to perform all the other analysis. First of all, we did a cluster analysis that highlighted the presence of 300 duplicated or closely related genotypes with identity more than 98%. So we excluded these duplicated individuals and ended up with 1,240 unique pitch accessions. In the plotting here, you see the dendrogram. Of course, you are not supposed to look closer in details of on it, but just to have the whole picture, where 3 clusters grouped on the red one, the oriental accessions coming from the Chinese collection, and other 2 were from occidental accessions, but were also divided into accession coming from modern breeding and those coming from traditional that were traditional accessions. This was the hint for the presence of population structure in the in our germplasm. Population structure is when the entire population is stratified into or more subpopulation that have different allele frequencies. So you can highlight the different subpopulations based on their allele frequencies, like in this picture where you see in Penela you see unstructured population. So in the 2 subpopulation allele frequencies are the same. This population is not structured, while in the panel B we have highly structured population with the 2 population clearly distinguished by the allele frequencies. To get and to analyze better analyze population structure, we used also a principal component analysis first, which divided our samples in the same three groups highlighted with the colored circles, the Oriental, Western traditional, and Western breeding. And to go in more details on this structure, we also ran a population stratification analysis through the algorithm present in the implemented in the software structure, which assigns for each individual, which is represented here as a vertical bar, based on the allele frequency, to a membership coefficient to one or the other subpopulation. The operator can superimpose the number of clusters of subgroups that can be detected and see the result. You see here that from the first with K2, so from the first round, we had the first division between the Western and Eastern accessions that originated from the long-term isolation of the evolutionary history of pitch. The third, at K3, the Western accession divided into Western from breeding and Western traditional, which reflect the genetic drift due to the founder effect that was the result of the choice of just a few parentals to develop the new varieties in the breeding programs that started in the 20th century. And then at K4, the Western breeding varieties divided into two other subgroups, grouping mostly pitch and from one hand and nectarines on the other. That are the two different types of fruits in peach, reflecting the separation between the breeding of the type peach and nectarine in the breeding history of peach. The optimal subdivision stratification was found at K3, so with the three groups from long-term and founder effect, while 665 individuals remained admitted. We also looked into the linkage disequilibrium in pitch, and we found high levels of LD in all chromosomes and in all subgroups. In all the three subgroups and also in the admixed group. 3 blocks of more than 1 to 2 megabases were present in all population, and here you see the representation, the heat map of the chromosome 4, where here are the length of the chromosome and the so the map of the chromosome 4 with the position of the SNPs. And here you see at the end, at the beginning, at the end of the chromosome, two blocks of high LD. This was probably this is attributable to artificial selection, while this was also observed to be a recombination called spot. So then we analyzed all this data through this software to perform a genome-wide association analysis. So we put together genotypic, phenotypic data, population structure, and also kinship matrix, which accounts for familial relatedness between accessions. Here are the results of this work. But it is just said that we chose to do 7 monogenic traits because this was the first attempt, the first time that GWAS was done in pitch after the genome sequencing of the species and the development of the array of SNP. And so we had the objective of ascertain the feasibility. So this was a proof of concept of the feasibility of genome-wide in pitch. So we chose 7 monogenic traits that are easy to score. That for which we already know the position in the genome, or for some of them also the gene was had been already isolated in the case of fruit pubescence and flesh color, and also okay, yeah, don't find it. The fruit pubescence and fresh color have already been isolated. We found significant association for all traits in already known genomic regions, but also we highlighted the need for SNP haplotypes to be more predictive than single marker in pitch due to the high LD. It's very important to say that phenotyping is crucial for a good genome-wide experiment result. In fact, the power of an association is highly dependent on the quality of the phenotypic data. And in this sense, the development and application of technical innovation, here are some of the a list of some of them available. Innovations for precisely and automatically phenotyping will certainly boost genetic diversity exploitation, like in experiments like genome-wide association analysis. The same set of accessions and markers, we decided at the end of the project, the Rubritomic project, to sorry, yes, to develop a core collection in pitch, because with the aim to use to develop the diversity of the pitch germplasm. Core collections are a subset of a larger germplasm collection containing the maximum possible genetic diversity with a minimum of repetitiveness. So we started from 1,200 accessions coming only from the European collections, because we could have problems in exchanging materials with Chinese colleagues. And we ended up with 169 accessions that represent the whole genome diversity of the initial collection. We started with a core collection of 69 accessions that were sorted out by the use of the software implementing the maximization strategy that compacts the genetic diversity into the minimum number of genotypes through the use only of molecular markers. And then we added individuals, 100 individuals, based on experts' evaluation of the breeding value, like economic, historical, traditional reason. We also try to conserve the population structure of the initial original collection. And we also added a controlled redundancy for reaching a more powerful power of significance in genetic studies. The pitch ref pop is also the accessions. To the accessions we also added 234 seedlings and cross parents, and these two type of materials made up the pitch ref pop, that is a multi-site collection, reference collection for pitch, that is replicated in 5 sites across Europe. You see here 2 in Spain, 2 in Italy, 1 is in at CREA in Rome, and 1 in Aarhus. Now I'm at the end of my talk, and I would like to stress still on the need for use, conserve and use the genetic resources. Conservation can be done in situ, so in their habitat, or ex situ conservation, like in seeds in germplasm banks. And we can also conserve in plant species, in fruit tree species. We conserve through ex situ in vivo, so planting and growing the plants in the field, like in germplasm collection. In CREA Rome, we have the National Fluid Germplasm Center, where we maintain, conserve more than 5,000 accessions belonging to 24 different cultivated fruit species. Here is a list, and 39 botanical species and interspecific hybrids. And we have in our experimental fields, we have germplasm collections, core collections, like the one of the pitch ref pop, and also parental populations, like the one that was as the reference for the prunus, the T by E, and those that were used also for the refinement of the genome sequence of pitch. Okay, now I'm done. Thanks for your attention. This is the intro of our institute in Rome. I only would like to mention my other colleagues that work from in Rome with me in the proteomics. Sorry, the Innovate project. Elisa Vendramin, Roberto Ciccoritti, and Ignazio Verde. I'm done, so if you have any questions, I'm available.